Hello, my name is Hayden Siesel, and I'm a sophomore in the College of Arts and Sciences at the University of Pennsylvania studying biology and health and societies. Thank you in advance for listening to this presentation on my summer research through PURM, and it took place in the Ridke Lab in the Perelman School of Medicine and was on the topic of investigating the signaling mechanism of GPER via BRET. A little overview about the project. It has been found that female sex in prior pregnancies have a protective effect on melanoma, and a recent Ridge Lab paper showed that this effect is likely due to estrogen signaling through the G protein coupled estrogen receptor, or GPER, on melanocytes. This receptor is thought to be GS coupled and cause CAMP activation of PKA, but as this has not yet been validated, my project was to try to determine the endogenous alpha subunit and consequently the signaling mechanism of GPER. In the background section, the first image shows the proposed mechanisms of the melanoma protective effect from past Ridke Lab research, which was based on the assumption GPER was GS coupled. Before I explain my project, I'll explain a bit more about G protein coupled receptors, or GPCRs, and bioluminescence resonance energy transfer, or BRET. GPCRs are the largest family of proteins targeted by approved drugs currently, and they are heterotrimeric with alpha, beta, and gamma subunits. When a ligand binds and the GPCR is activated, the alpha subunit dissociates from the beta gamma complex, and primarily the alpha subunit is then involved in creating a signaling cascade. BRET works by transfecting the alpha subunit with R-luciferase and the beta subunit with green fluorescent protein, or GFP, two proteins that emit different wavelengths of light. Luciferase's substrate colon-tyrosine is added, activating luciferase, causing it to produce light. The closer GFP is, the higher the GFP activation is, since luciferase donates photons to GFP when it emits light. Therefore, the ratio between luciferase activity and GFP activity, determined via spectroscopy and being called net BRET when compared to the baseline ratio, tells the GPCR's activity levels. This can be used to determine ligand and alpha subunit properties, among other questions. A diagram of how BRET works is in the bottom corner of background, if you would prefer a visual interpretation. About my methods, I performed restriction enzyme cloning of both wild type and P16L GPER into a PRRL backbone. This was not relevant to my project specifically, but having these plasmids will be helpful in future lab endeavors, specifically because the P16L mutation may negatively affect the protective effect and needs to be studied for personalized medicine reasons. Below, there is also a general BRET protocol of the Ridke Lab, which I use to perform all of my BRET studies. Moving on to data and results, the first graph is of a BRET I performed using ADRB2 and the associated ligand, which is isoproterenol. And this was a control in the paper we used to determine our BRET methodology. And so the different levels of ligand of isoproterenol showed that SS and SL seemed to have what was closest to a dose response curve. And you can see they also had the greatest response, as opposed to the other alpha subunits having sort of random variation around the zero net BRET. And so that shows that SS and SL are likely the endogenous alpha subunits. Moving on to the table, I performed a BRET with 100 nanomolar LNS, which is the ligand for GPER. And this BRET had all of the different alpha subunits and GPER transfected into the cells. And in this, we see that G13 was shown to possibly be the endogenous subunit of GPER because it had the most negative net BRET. And then I performed a BRET going off of this with GPER and different levels of LNS. And only the top few alpha subunits to help determine the endogenous receptor. And again, G13, if you look, had the greatest response and most like a dose curve. And so this led me to believe that G13 was likely the endogenous alpha subunit of GPER. So in conclusion, in future assays, we must determine if G13 is truly the endogenous alpha subunit of GPER. Confirming this or rejecting this hypothesis and determining there is another endogenous alpha subunit and signaling mechanism will allow for improved treatments for melanoma and other somatic cancers. Therefore, this project and future projects related to it could help improve the standard of care for melanoma and other related cancers. Thank you so much to the Ridke Lab and Kerr for supporting me in this project this summer, as I have learned a lot and am excited for my future in research. Thank you all again for listening to my presentation, and I hope that you have a great day. Thank you.